So this is the briefing for practical 3, that is photosynthesis. Again, can't do it on in the laboratory yet, so have no choice to conduct a briefing through this recording to help you to prepare the report. Let me start. So this is on photosynthesis. It is involving the metabolic pathway of photosynthesis. Will be also covered in theory. Basically, photosynthesis involve two types of pathway, light reaction pathway as well as light independent pathway or Kelvin cycle. The light reaction pathway is involving certain carriers of electron. Once it receives the photon from the sun, and these carriers will transfer electron until it reaches water to break it into oxygen, to simplify the whole thing. Well, in between these carriers you can incorporate the DCPIP, which once it receives electron, it will change in terms of color from dark blue, and as much as it receives electron, it changes to no color, dark blue to colorless. So this is explained in the Hill reaction and can be utilized to study the photosynthesis reaction rate. And in terms of this practical, there are two parts. Part 1 is to extract the chlorophyll, the pigment, using ethanol from the leaf, and then looking at understanding the maximum absorption wavelength of the pigment, and you will realize it will absorb maximum at ultraviolet blue as well as red visible range. So that's what we're going to do in part 1 by using a spectrophotometer to study its absorption wavelength, maximal absorption wavelength. And then part 2, we're going to take the spinach leaf again and try to extract the chloroplast. And once the chloroplast, which is a living organelle, is extracted, we leave it in ice throughout the experiment and use it bit by bit to study its reaction rate for determining, for instance, whether light is important, one of the experiment, and also the uncoupler, and also another chemical, gramicidine, as well as DCMU, in terms their contribution to the reaction rate. So consider I have already given an overview on this practical photosynthesis. Let's move on in how we are going to perform the experiments. And since we can't do the experiment in the laboratory, there is for part 1 3 videos I have managed to obtain, and for part 2 2 videos. Try to watch these videos to understand. For instance, the first video, which is part 1 of part 2, it explains about the chlorophyll extract. Although it's been like home setting, I'm not sure whether you can do it in your house, but if you can, go ahead. But it is not a necessary or compulsory thing to conduct it and actually provide it in the report. It is just for sake of understanding the extraction of chlorophyll. And then you have also part 2, continuation of the first part, and looking into explaining further on the pigment of chlorophyll. And the third video is more on chlorophyll extract, and it also demonstrates the change in color, that is chlorophyll, once it's exposed to UV light. So again, you may do this at home, but you need a certain specialized apparatus for UV light. Maybe you can use a fake money detector if you have, or a fly trap UV light also can be utilized in this home experiment. But again, it's optional. It's not necessary for you to conduct it. For part 2, which is involving the chloroplast extract, there are two videos. Here, Hill reaction is explained in detail about the DCPIP able to absorb the electrons and change color from dark blue to colorless. And then another interesting thing about this video, it is like a home setting, but still perhaps need to be done in the laboratory. Still have certain things impossible to be done at home, although the video describes it like you can do it at home. And then you also have the extraction of chloroplast, and using spectrophotometer to study it, and also to the extent of using DCMU. So this video is the best, very close to our experiment. So please ensure, if you really must watch one video, this is the video that you should watch to understand what we are doing in part 2. Right. I also have incorporated a home experiment, since you are unable to come to the lab. So this one is interesting, where you can do this in the house. You need a potted plant, any potted plant with green leaves. Okay, and then also need alcohol. This you can get from any pharmacy. Ask for isopropyl alcohol or rubbing alcohol, which is used for first aid, I assume, or cleaning wounds. And then you also have iodine solution, also for first aid, for wound dressing. So these two items should not have any problem in obtaining, and in fact your seniors have got this easily from the pharmacy, or other shops or health shop that sells them. And then piece of paper to cover the leaf, as well as water. Okay, so all able to be achieved, or therefore can do this experiment. So what you need to do is, you need to keep the potted plant away from light in the dark for 48 hours. And after that, you you take the potted plant and cover it as shown in figure. However, I have made it a bit more challenging, where I want the shape of the cover, or if you want it to be clear, it's up to you, either visible or opaque area, to be having this V shape. So let me try to draw to you. So hopefully somehow you can figure out to come up with a shape such as this, and therefore uh, you have covered it. And then what you need to do is now leave the plant in sunlight, but I didn't mention for how long. uh, so you have to figure out for how long you need to leave in light. Therefore, photosynthesis can be said to have minimally completed. And after that, pluck the leaf and test it for starch. And I have given a YouTube video on how to test using iodine for starch. Okay. Hopefully you get to see what is found on the leaf that has been covered with a V shape and report it in your laboratory report. Okay, and next yeah, for sure we cannot do the laboratory experiment that we intend to do, that part 1 and part 2, but it involves light reaction, and light reaction has two photo systems, PS1 and PS2. With the PS2 and PS1, there will be electron transfers, and what happens is uh, it moves the electron from these carriers that is found until reaches water, and water gets oxidized. Okay, so uh, if the electron is excited and it can move through the carrier until to water, it is therefore possible we put the DCPIP, which can receive the electron, and when it receives electron, it changes color, and we can therefore measure these changes of color using spectrophotometer, and that's what we are doing. So let's move to the experiment itself, because I don't want the briefing to be very long, just short and enough for you to understand about both part 1 as well as part 2. So part 1, we're going to look into extracting the chlorophyll, and then by using the spectrophotometer, by changing various wavelength, um, 350 I think up to 900, every time change 5 350 to 355 through 360 until 900, and see where it absorbs the maximum. Okay, theoretically it's supposed to be absorbing maximum at ultraviolet blue, as well as red region of the range of wavelength. Okay, and then we're going to look into the Hill reaction, where reduction method is applied, where we're going to look into the reduction of DCPIP. Is there, more exposure given to the chloroplast extract? Will it cause more of the DCPIP to change color? So that's what we are intending to see. Okay, and then we're also going to see whether there is important in light intensity for the rate of photosynthesis. This is followed by what happens if we increase the chloroplast concentration from low to high, whether it has any effect to the rate of photosynthesis, and also in terms of DCMU and gramicidin. We're going to look into an inhibitor, what it inhibits, go and find out. Okay, and then gramicidin as an uncoupler, it basically creates a gap in the electron transfer, and what happens after that to the rate of photosynthesis. So please identify by actually uh, using the hypothetical results that I will provide to you, since you cannot come to the laboratory. Okay, so let's start the chlorophyll extract experiment, where we need to prepare chlorophyll extract. And how do we do that? We actually take several spinach leaf. So again, I have to draw all this. So what we're going to do is, have a beaker which has been set up on. Okay, this is the hot plate. Assume this is the hot plate. The best I can do. Okay, has a wire to the socket and a knob. Okay, So here you put the leaf, few complete leaf, right? Then you also put ethanol, just enough to submerge the leaf, and then set it at low temperature, maybe 1 or 2, okay, and hopefully heat comes in. Okay, so it says here carefully immerse the leaf in ethanol, and then on a hot plate slowly heat, but make sure the leaves are not crushed, so complete leaf will be good. And then make sure the ethanol does not boil, okay, just enough to warm the leaf and slowly leach out the chlorophyll. That is what we are trying to do. So we do this for some time because no proper time has been given. Perhaps by looking at the chlorophyll exiting out until the green color of the ethanol is coming about. So ethanol is actually a clear solution. Once it start to leach out the chlorophyll, the ethanol will start to become green in color. That is the indicator. Okay. So assume you're already satisfied. Take out the extract and you filter it, okay, by putting a filter paper. You put a filter paper here, okay, and pour the thing. Okay. Hopefully you get to filter it, okay, and then according to the next step, you pour it into, sorry about that, okay, again let me draw a cuvette. Okay, this is the cuvette. You pour up to 3/4 full, and somewhere here, no need to be accurate, because as long the light passes below with the liquid present is sufficient. No need to be, have to be, 2 ml or 2.5 ml. Just pour accordingly the chlorophyll extract here. So the chlorophyll extract is already found within the cuvette. Take the cuvette and go to the spectrophotometer and read the absorbance. Once the the wavelength that's been set is 750, so you set the spectrophotometer to 750, and also they have mentioned you have to blank the spectrophotometer using ethanol. This step is a necessity to make the so-called needle of the spectrophotometer to become zero. Okay, so that's the use of the blank. And then once it's ready, reading is zero, then put this cuvette that has the chlorophyll extract and start reading, and this will be the measurement. Hopefully the measurement is reached the target reading of 0.009 to 0.1. If say you have over extracted, say it became zero point 0.2, then you need to perhaps dilute 2 times and then read again until it is in between this range. So this step is just to get sufficient amount of chlorophyll extract. Okay, next step is the one that we're going to look into, where the chlorophyll extract absorb maximally. The range of chlorophyll extract can absorb. Okay, so we reset the spectrophotometer to 350. Okay, this is the sorry, shouldn't be capital. So assuming we set it at this wavelength, and then again we have to blank the instrument to be 0, and then again we put this cuvette which has the chlorophyll extract into the slot of the spectrophotometer, and then start reading. So you should have the absorbance reading determined. Okay, next you change the absorbance the wavelength from 350 to 355. Okay, and then blank the instrument and read. You do this like every time 5 increment, and read 365 and so on until you reach 900. So quite a lot of hard work. You must decide and you must find out all the absorbance reading from 350 to 900. But there is a faster route to achieving this by using a particular feature of the spectrophotometer, where it can continuously read from 350 to 900 until can even draw the spectrum. But for this purpose, follow the instruction so that I don't confuse you further. But bear in mind there's also another feature that can read continuously and get the result within a few seconds. Okay, so assuming you already get the 350 to 900 absorbance, here they say plot a graph, your wavelength versus absorbance. So assume this is the graph. Okay, this is wavelength, and this is absorbance, and then therefore how will it be like? Is it going to be like this with different maximal absorption, or simply said there should be two particular region of maximum absorption? That I will let you to decide after I give you the hypothetical result. Draw the graph and find out. Okay. The next part is part 2, that is the Hill reaction using this dye reduction method. And it has been said it's a very old discovery by this guy in 1930s, where he realized that once the chloroplast or thylakoid isolate uh, is able to receive the electron, and therefore there will be loss of this has been explained here. Okay, accepts electron, DCPIP, which stands for dichlorophenol idophenol. So it accepts electron, become reduced, and changes color from blue, which is the oxidized state of the DCPIP, to become colorless. And this been described here. Okay, from blue, because of the photosynthesis, the light reaction pathway, it become colorless. Okay, because DCPIP can be taking the electron that flows to water in between, and therefore can become colorless. Okay, right. So we can use this reaction of Hill, and with the provided we isolate chloroplast, and then we can study using these two in terms of whether this one's supposed to be chloroplast. We can study whether the effect of increasing the plus concentration has any effect on the reaction rate, whether light has any effect on photosynthesis, whether the herbicide DCMU as well as gramicidin either on their own or combined have any effect on the effect of DCPIP or reaction rate. Okay, and another thing they describe here, oxidized DCPIP will absorb maximum at 600, so you have to adjust the spectrophotometer to 600 nm. Well, there is also preparation or pre-practical session, but just have to explain here. For buffer, you also need a buffer because chlorophyll extract need to be ensured does not have change in pH, which can destroy its function. So we normally use a phosphate buffer solution. It's called in short PBS, phosphate buffer solution, and we buy it as a tablet and just put following the instruction, put maybe 1 liter of water, and there it's already prepared after dissolving. Okay, and then chloroplast or thylakoid extract is done this way. Again, I don't have a video. We are working out on a video how it is done, so we will upload it when the time comes. I can only attempt to draw it now. So take the spinach leaf, for each group maybe this much, 15 to 20 gram, but when we conduct it, we maybe 5 groups, so we multiply it for 5 groups and everything we multiply. Okay, so what we do, we take these leaves. Now we have to use a blender, so hopefully I can draw a blender. Okay, it's called a Waring blender, quite a unique one, but we are only using more of a household blender. Okay, so assuming this is the blender, we put the leaf as much as been specified by weighing, and then put the buffer. Okay, right, so assuming the leaves are here and inside the buffer already, we cover it with a cover and put it into 30 seconds, probably a pulse blendering only, for 30 seconds only, very fast, done. And then what we do is we put again filter funnel. Okay, we use this cheesecloth. Just Google it, you will see what's how the cheesecloth looks like. This is a big funnel. We try to put the cheesecloth and then pour the content after blendering for just 30 seconds. Hopefully the liquid comes out quick enough, and then we collect in a beaker. But we need to store the extract from then onwards into so let me clear some of the space here to redraw. Okay, so we have to keep it in ice. How do we do that is this is they call it filtrate. We put a bucket here, and then with lots of ice cubes or mashed ice. In the lab there is an ice maker, so we just put it there, crush ice. So this is the filtrate and it has the chloroplast extract. So what do we do next? You can take care of this all from here, and then next we have to ensure we prepare DCPIP, DCMU, gramicidin. This is where you have to use this formula. okay, or formulas, okay, where n is equals to, okay, I'm not going to go in detail because there's another video for that you would have seen by now. Okay, now we're going to do the reduction of DCPIP and measure the photosynthesis synthetic rate. Okay, so this is our preparation, and then you need to pour the chloroplast extract into a cuvette. Okay, so clear this, and then you pour it into a cuvette. It says here put into a cuvette up to 3/4 full. Okay, don't need to be accurate. Put one drop, very tiny drop, to make it faint color change. Normally to mix it we cover with the parafilm. I have to show you what is a parafilm, and then we tilt it 180 degrees upside down a few times, then it will be mixed. Okay, right, so set the spectrophotometer at 600 nm, and then read the reading. So at 600, what is the reading? This is referred as T equals 0 time. Okay? And then place the cuvette in a small beaker of water 50 cm from the 100-watt desk. Okay, I have to show you again by drawing. Let me clear all these, hopefully, and that also. So you have this cuvette with the chloroplast extract. Now you need to, make it have light for photosynthesis to take place. And the best I can draw light. Okay. And then in the lab there will be, very bright. So we put this whole thing in a box. Okay, cover in a cardboard box which is concealed from light from the outside environment. Okay, and leave it there for 5 minutes exactly. Okay, after 5 minutes we take it out from the box, go to the spectrophotometer, and again do what's been done for the zero time. Read it at 600 nm, and we do this every time 5 minutes. Okay, until the DCPIP supposedly fades. So what will happen to the absorbance? Okay, yeah, so we expose it for 5 minutes and then go and read the measurement in the spectrophotometer, and then another 5 minutes, read it exposed to the light. Then this distance of the light must be as indicated 50 cm. Okay, so every time 5 minutes, probably we will do it until 30 minutes. Okay, so 5 and then read, and then record the absorbance. So what will happen to the absorbance? Is it going up or is it coming down? You will get the hypothetical result, explain the outcome. Okay, and then we're going to look into effect of light. For this we're going to use 4 cuvettes. Okay, and we do like this to the 4 cuvettes. Looks like chloroplast extract is there for everything except for this, don't have. And then this buffer is just to make sure the n is 2.5. Okay, and then here no DCPIP, and then this one is interesting. Put aluminium foil all outside of the cuvette with the chloroplast extract, therefore the light cannot penetrate even though you keep under the lamp. Okay, so do this for the 4 cuvettes, and then again put it into light source, expose it for first one 0 minute, and read at 600, no exposure to the light, and then expose to 1 minute of light, and then read all. Of course, this one number 4, take out the aluminium foil, take out aluminum foil and then only read in the spectrophotometer, then immediately put back the aluminium foil for the experiment. Okay, even inside this light source must have the aluminium foil for cuvette 4 alone. So expose it 2 minute, the total time 2 minute, 4 minute, and then 8 minute, and then get all the reading and compare. Is there a reason for having light in the photosynthesis? What happens if exposure is longer? What happens if no chloroplast? What happens if there is no DCPIP? So explain in the results section, right? And then effect of chloroplast concentration on the rate of photosynthesis. What happens if we firstly no chloroplast extract, and then what happens if we increase the chloroplast concentration from 0.5 until 1.5? Still must have DCPIP for all, and then exposure, initially no exposure until 8 minutes of exposure in this setup here using the cardboard box with the bulb. Okay, so that is on chloroplast concentration, followed by another one is DCMU concentration on the rate of photosynthesis. For these, we have two different concentration: low concentration of DCMU and then high concentration of DCMU. So one of it put the low concentration, another one put slightly higher concentration, and then everything else you have, DCPIP also have, again exposed to the cardboard box with the light source, 1 minute, 2 minute, 4 minute, 8 minute, and then from the initial compare it, what happens in terms of the presence of DCMU at low concentration and also high concentration. What does it do to the reaction rate? Okay, and one more is gramicidin. This one we also have the DCMU. First cuvette, both also don't have. Okay. Second cuvette have gramicidin, and then third cuvette both gramicidin and DCMU. So what does one does to the other when it's combined? Again, do it. Supposed to do the exposure to the light source for 1 minute, 2 minute, 4 minute, 8 minute, and then compare the reaction rate compared to having gramicidin alone, having DCMU alone, and also having in combination of gramicidin and DCMU, okay, as well as not having at all. So this icon here, again, it's online learning. You are to perform activities suggested to you at home. Okay, please do that and write a short report. So must have a report with result achieved. Probably have to give a snapshot on the outcome. Okay, watch the video given to you and write report based on hypothetical result that I will give you on part 1 as well as part 2. And then as usual, I prepared some question discussed on the result obtained for the home experiment. From the hypothetical result, talk about whether light intensity, chloroplast concentration, the inhibitor and uncoupler compound affect the rate of photosynthesis. And then principles on DCPIP, gramicidin, DCMU also can provide at the results and discussion sections. And also lastly, explain why need to place the cuvette in the beaker of water. Yeah, looks like I have uh, not shown that one. Let me correct myself. Okay, one thing I have left out is, uh, let me correct it, the drawing. Okay, one is they actually ask you, let me do it correctly. They actually ask you to put this in a beaker. Okay, oh, not a very good drawing there. Okay, okay, let me just draw a small beaker. Okay, so they ask you to put water to set the beaker, and only then have the box. Okay, there is actually water being put and the beaker has been set on it. Okay, if just to they should have it somewhere. Yeah, a small beaker of water of 50 cm from. Okay, so this is the one I overlooked just now. So now place the cuvette in a small beaker of water. Okay, so what is the purpose of the beaker with water? You answer. Okay, so also ask you that question. Okay, that's it. I have finished with this briefing that is meant for the practical 3, that is photosynthesis. Thank you very much.